According to the United States Department of Agriculture, Typha latifolia, or broadleaf cattail, is a native species to the continental United States and Canada, but it has become a prominent invasive species in regions which has been introduced to, including regions as geographically separated as the Philippines and Hawaii. Whether invasive or native to a region and spreading to disturbed habitats, due to its ability to rapidly colonize habitats, Typha tends to monopolize vegetation and exclude wetland flora. As Typha latifolia is an emergent plant species and often spends a portion of its life partially submerged in water, understanding the limits of the species' tolerance for factors related to water quality, such as water temperature, dissolved oxygen, and pH, is critical to management and control of the species.
Dissolved oxygen is known as the amount of available oxygen dissolved in a body of water molecularly. The amount of dissolved oxygen is often a strong indicator of the health of many aquatic ecosystems. Low dissolved oxygen levels generally indicate pollution, often caused by an influx of nutrients, eutrophication, and respiring decomposers. Low dissolved oxygen levels can inhibit a variety of organisms from growing or thriving in a habitat. In bodies of water that have higher amounts of dissolved oxygen, we often find clear waters and higher biodiversity.
pH is a measure of the concentration of hydrogen ions in the water. Because enzymes function best in a narrow range of pH, pH can have a large impact on the growth of organisms.
Previous laboratory research has found that pH does not appear to have an effect on cattail germination and growth, with cattails showing tolerance for a broad range of pH conditions, as acidic as 4 moles per liter and as basic as 12 moles per liter. Though Typha latifolia appears to be able to tolerate a broad range of pH conditions, previous research has suggested its tolerance range for dissolved oxygen levels may be more narrow. A previous study focusing on germination rates and early growth of Typha latifolia plants found that Typha latifolia's growth may be limited by high levels of dissolved oxygen.
Our hypothesis states that Typha latifolia grows better in environments where there is a lower amount of dissolved oxygen in the surrounding water. As the shoot, also known as the fruiting spike, is a reproductive structure of the plant, the size, height, and width of the plant is understood to serve as a reasonable proxy measurement of overall plant growth. For the purposes of this experiment, then, we expect that Typha latifolia in environments with lower levels of dissolved oxygen in the water will have taller and wider shoots. For our experiment, we traveled to 4 different locations that displayed evidence of cattail populations. We first measured the pH, DO, and temperature of a water sample at each location. Then we measured the height and width of the shoot of up to 20 cattails, or as many were available, using a meter stick and recorded our results. We also recorded the color of the cattails and whether each cattail was emergent, submerged, or dry. We then recorded weather, temperature, and canopy cover. In addition to our quantitative data, we also recorded observations of the surrounding habitat.
Our data shows the average length of the shoots being greatest at Lake Shenandoah, which contained the highest pH of 7.53 and dissolved oxygen levels at 11.7 milligrams per liter. The average width of the shoots in comparison with dissolved oxygen shows the width being greatest at Moortown Pond, containing dissolved oxygen levels of 9.9 milligrams per liter. The average width of the shoots in comparison with pH shows the width being greatest also at Moortown Pond, containing a pH of 6.74. There appears to be a fair amount of hedging due to limitations. Some of the limitations we encountered include low sample sizes in each location, limited accessibility and availability both legally and locally, thus could not ensure we were visiting sites with distinct differences in dissolved oxygen levels, as well as a limited time frame preventing us from taking multiple measurements of water quality and overall cattail growth. We were also limited by the season in which many cattails had begun to die and burst open, thus possibly affecting length and width measurements. In the literature, Typhia latifolia grows best in low-dissolved oxygen environments, where numerous other species are limited by their inability to tolerate the low oxygen conditions. However, it is capable of growing in high-dissolved oxygen environments, but seems to do poorly. It is possible that the Typhia latifolia is a poor competitor and can only persist in the absence of competitive species. Due to limitations, we urge caution and suggest more rigorous research with a longer time span and additional resources to circumvent the practical and temporal limitations we encountered in our study.